All right, I want to thank everyone for coming today. It's a real pleasure to have Dr. Alvis here. Dr. Alvis is actually a part of WIU. She holds appointments in anthropology and zoology and chemistry. And you might have noticed she's just down the hallway and has a lab here too. A lot of her research is biology related. And so I thought it's more important than ever to start having more interdisciplinary approaches across departments. I'm hoping in the future some students might be interested in working with Dr. Alvis in some capacity, whether it be a collaboration or maybe an honors project or who knows. But you'll find out real quickly how related research is to our department. Let's see. So let me just give you a quick background that I found on her website. Dr. Alvis earned her bachelor's degree in anthropology from the University of Washington with an emphasis in archaeology and an undergraduate research focus on the analysis of skeletal remains and geoarchaeological deposits. She received her PhD in anthropology with a minor in human genetics from the University of Minnesota, investigating factors that influence the preservation and detection of DNA in ancient and forensic specimens. In addition to her academic experience, Dr. Alvis worked in several years as a forensic scientist in DNA biology, so that might be something else to think about in our department. With a section of the Minnesota Bureau of Criminal Apprehension Forensic Science Laboratory. She led the WIU Archaeological Field School from 2017 to 2021, and also conducted archaeological excavations in Israel, South Africa, and throughout the Midwestern United States. Thank you so much for coming and visiting us.
Thank you so much for having me. All right, I am excited to be here and talk to you. I actually haven't ever given a presentation to this department before, even though this is my 12th year here, so I'm excited. All right, you can see there's a lot of stuff in the title, right? And my goal here is to sort of introduce myself to those of you who don't really know me, and then talk about some of the different research projects I've been doing here, and the project that's just getting underway. So I'm going to keep an eye on my clock, and I will rush forward to the most recent stuff if I start to run out of time. And I need to use the. Yes, no.
Maybe the easiest thing I just for now, just use the pointer and just click the.
Oh, over here. Okay.
This easiest way. I don't know if there's another one.
That works for me. Okay, so I'm an anthropologist. That's my primary title, and a lot of people associate anthropology with just the social sciences, because in a lot of places that where you're taking a general education course in anthropology, that might be what you're talking about. But anthropology really has 4 main fields. Okay, we study the full scope of human diversity through space and time. Okay, I was actually a double major as an undergraduate in environmental. I was going to pursue it in environmental science, Bachelor of Science, and then also a BA in international studies. And I did that for 2 years before I discovered anthropology and realized that I could take fewer courses and still study all the same stuff. So it was good background, really in natural science approaches to things in the environmental science program. And I took that with me when I moved to anthropology. But you can see that anthropologists study culture of modern peoples, archaeology of ancient peoples, and then linguistic or cognitive areas about being human, and our biology. Okay, and my areas are kind of overlapping most of the right side of that Venn diagram. For the most part of the first part of my education and career, I focused on what's called taphonomy, which is the study of what happens to things after they die. Okay, and so for my senior years in junior and senior year in my undergraduate, I worked in a lab where I was doing skeletal analysis and study of soils and sediments from ancient deposits, which is geoarchaeology. I also worked for 2 summers on a early Bronze Age site in Israel. It was an outpost of the first Egyptian empire, the Pharaoh Narmer's, who was one of his outposts, and we know that because of little the carvings you see on the pottery, and this is like a cylinder seal that they would have rolled into clay, and then it would have made the impression of this name. And I worked for 4 years after my bachelor's degree as a professional archaeologist in the Midwestern United States. And again, when I was in the winter times, when I would be in the laboratory, I was focusing on the analysis of skeletal remains, mostly animal bones, and paleoethnobotany, which is the study of plant remains in archaeological context. Okay. But I started getting really interested in the new and emerging field of ancient DNA at that time. And I ended up deciding to go to graduate school to study that. So I went to the University of Minnesota, and my PhD was focused on both biological anthropology and archaeology questions. And I did a graduate minor in human genetics, and everyone else in the graduate minor were medical students that were going to pursue a human genetics area of medicine. So that was a really interesting thing to be doing simultaneously, and getting different, perspectives on genetics in ancient contexts and what we know about DNA and genetics today. And my question ended up being, what is it actually that we can, how can we predict what is going to be a good sample to send in? And when we get it in the laboratory, what strategies would be the best ways to use that, to use to address the, or to get the most information out of a sample. I also spent many semesters teaching human evolution course, which was a big biology gen ed course. And I taught a forensic and molecular anthropology class as a graduate instructor. So I realized that when I'm looking at DNA taphonomy, which is what happens to DNA after something dies, right? There's a lot of variables, and I grouped them into variables about the environment that the sample is exposed to, variables about the specimen itself. So is it plant remains? Is it a bone? How big is the bone? Is it a blood stain? What is the specimen? And then what kind of lab strategies are available? And are there, how do we choose which ones to use for extracting the DNA, for contamination control, for amplification targets, for purifying our DNA, all those kinds of things. So those are the questions. I got samples from where my professors then were working. So I went to South Africa. I collected bone and soil samples from the really old, this was kind of a long shot, but we were going to excavate there. So 1.2 million year old hominin site of prom dry. And I didn't take hominin bones, but I took some other animal bone for testing in the laboratory, and then soil samples. Wonderwork Cave goes back to, it was continuously occupied for about 400,000 years, up until 1910, when a farmer lived there with his family while they were building their house. And so it has many layers, really nicely preserved strata that have all kinds of evidence of human occupation. And so I got plant remains and animal bone from that, from the various layers at that site, and some soil samples. Border Cave is another big, one of the early anatomically modern human sites. Goes back to about 200,000 years ago. It's way up high on a cliff. And so really there's almost nothing in there that wasn't brought in by birds or the humans that climbed up there. And one of the earliest possible calendars is this Lubumbo bone. I did not do testing on that, but I tested plant remains from this site and soil samples. And I also helped with an archaeological field school in Minnesota, because it's a village about 900 years old. And they have really acidic soil there, so there's really bad bone preservation. And so most of the bones that they were finding were just like really fragmentary, and you couldn't tell by the morphology or the shape of them what kind of animals they came from. So I took a lot of those bones, and I had the students collect adjacent soil samples as they were digging that whole summer. And so I took the bone fragments, and took them into the laboratory to identify the species on those, to find out what people were eating at that site 900 years ago. So this is where I went to do the ancient DNA work. It's the Paleo DNA Laboratory at Lakehead University in Canada, which is, it's not far from Minnesota where I was then. The building was built specifically for ancient DNA analysis. So they like UV irradiated the drywall as they were putting it up. I mean, they went like all out to make sure that there was no, there was no DNA on, stray DNA anywhere when that place opened. And you had to get in the full hazmat suit. There are different rooms for different parts of the process. And you'd have to go through an air shower, which is like you get locked in a little room and it would like blow air on you. It's sort of like being in an elevator, but it's just blowing air. And there's vacuums around the floor that like suck any like hairs or cells or anything that might have been stuck to your hazmat suit. It sucks those away before you go to the next room. And so that was pretty, I did an internship there, and then I went back 2 summers to do my own lab research in that facility. I also realized that a lot of the questions I was asking about DNA preservation were being asked by the forensic community. A lot of the literature was coming from them. And so I found out that there was a summer internship program at the university, or at the Minnesota Bureau of Criminal Apprehension, which is a state forensic laboratory in Minnesota. And so I applied for that while I was a grad student. Most of the applicants are undergrads, but I applied for that and got a summer internship there. And I asked questions about, or my research project was questions about the protocols and contamination controls that are used in the forensic lab. And they had some contamination problems where like different samples from different cases had cross-contaminated, really bad stuff you don't want to ever have happen. And I got to the root of some of that. Some of the protocols they were using weren't consistent, or 2 different devices that are supposed to do the same thing don't actually work as well. Don't both work equally well. So we were able to modify the protocols used in that lab from my internship. And then they encouraged me to apply for a job there, just as with budget issues, the teaching assistant positions were disappearing in my graduate program. So I decided I worked full-time as a forensic DNA analyst for several years while I was writing out my dissertation. And that work that I did for my internship was a chapter of my dissertation. So this is what I dealt with. Okay, I had all these variables, and I started having to think about how are you going to get any useful information when you have so many possible variables. And so I learned about how to use a relational database and access. And so each of these things you see here, each box is a spreadsheet, like an Excel type spreadsheet. And what you see are the column headings, and then they're connected to each other in different ways. Because it was a really complicated question, right? If I have a certain set of samples, how do I decide which ones to send in? It cost at the time about $1,000 per sample to try. And if you couldn't, if it didn't work, then you were out of luck, okay? So I got good at figuring out or tackling really complex problems like that. And I've used that in other ways, as you'll see in my research, that mindset or ability going forward. So I finished my PhD in 2012, joined the anthropology faculty here as a temporary instructor in 2013. And then in 2014, I started a joint appointment with chemistry because they have a forensic chemistry program, and their forensic chemistry person took a job somewhere else. And suddenly they desperately needed someone with forensic experience, and I just happened to be there. So I had, I'd been asked to, one of the things that their students are required to do is 3 semester hours of lab research for a senior project kind of lab research. And so they needed someone to do that. So I did my best to develop projects that would be, semester-long projects that students could do with an interest in forensics. They also, over the 11 years that I taught here previously now, I've taught more than 25 different courses across different, these different fields. Okay. I don't know. I mean, it's interesting stuff, but it's a lot of course prep. And I don't try to put them on a slide anymore, but there's everything that you're seeing here going forward from this point. If you want more information, talks, presentations, and my teaching stuff and everything is on my website, which is mylastname.com. So I established these research sites. They had no lab space to give me where I could do the kind of DNA work that I trained to do. And so I had to get creative, and come up with these projects for undergrads. And so I rented some different plots of lands around Western Illinois. And I had the students do soil and water profiling. So if you had a shovel in somebody's trunk, in a suspect's trunk, and you had a crime scene, how would you tell if that dirt that was on the shovel matched the crime scene? Those kinds of questions. Biochemistry, histamine analysis, which we'll talk about in a minute. And I had anthropology students who wanted to do lab work too. So we had stuff with them. And then the coming soon stuff is the social amoeba molecular biology work that we're going to talk about at the end. So here's some of the kinds of research project topics that people looked at. Here's one of our trail cameras with a possum chewing on a cow leg bone. You can see that there's a little sticky trap. One of the students was doing an entomology study, so we had sticky traps that he'd go out and collect every once in a while. And we did all kinds of analysis using various equipment in chemistry. So here's one of our early lessons. If you leave a meaty rack of ribs in front of your camera, a vulture will take it and you can walk for miles and we'll never find any trace of those bones. Okay, so we realized that we had to we actually started drilling a hole through the cow leg bone and then putting a chain around a tree. So it would stay in front of the camera, but we could still see different animals visiting and look at the kind of markings they were making on the bones. So we had a lot of different animals. I only included a few here. Deer are very curious. As soon as you put out a camera, a deer will show up within 24 hours and be sniffing around. Fox, we buried some bones. They dug them up. We had some stuff down the river, some visitors down there. I still don't know exactly what that is. And I'm curious because its face is kind of obscured by the grass. And then we had primates. Okay, so this is a pair of my students who had to go out every week and collect data off of their samples or their bones that they left out there. And they decided to show up on Halloween wearing a horse mask. And then they waited for a really long time for me to actually look at the pictures before, they're like, have you looked at the pictures yet? Then I found out why. So another area that I got into then was looking at histamine. Okay, histamine is something that our body produces, but it's also something that bacteria produce when something is decomposing. And so there are a lot of potential research questions in forensics that might potentially be asked or be answered by taking, a quantitation of how much histamine is in some kind of a sample, and then we'd have to factor for things like temperature and whatever. But we were looking at if that would be possible, and could we develop a test kit. The time since disappearance is like if there's a bowl of chili left on on the person's table and they're gone and nobody knows what happened to them. How long has the chili been sitting out? Those kinds of questions as well. There are also histamine disorders. Okay, rosacea is one. So if your body already has, you already have a high level of natural histamine in your body, and then you eat something that has a lot of histamine in it, it can cause problems for certain people. So rosacea is actually something that I have a little bit of. It causes red skin on the face. Migraine headaches is another thing that can be associated with histamine. Attention and behavioral disorders, and mast cell disorders. So mast cells in our body actually store histamine, okay? And then they release histamine when needed, like if we get a cut, they'll release histamine and it will help to facilitate the healing process to start. But if people with the disorders like mastocytosis, okay, like you see here, people that have this disorder, if they eat the wrong things or experience the wrong kind of situation, then they will break out in these blisters. And so there are concerns for families with these problems about how to manage it, right? How to live with these conditions. And another one that is indicated that might be related to mast cell and histamine issues is neurofibromatosis. So neurofibromatosis, and there are three types. I'm talking about type 1 here, which is the most common one. Is it autosomal dominant genetic disorder? So you have two copies of the gene, one from each of your parents. And if one of those genes is not functioning properly, okay, if one copy is not functioning properly, then you get this effect, okay? And so here on the top, we have a girl and her dad. They both have the disorder. You can see that his has progressed to having lots and lots of benign tumors. And her skin is looking a little rough, but not so serious yet, okay? The folks on the bottom have another kind of tumor that can occur with this disorder, which is called a plexiform tumor. In both cases, the tumors arise from the nerve sheath, okay? And anywhere in your body that you have nerves, the tumors can happen, okay? In the case of the plexiforms, it's like the tumor grows along like a whole network of nerves. And then the other ones are just like little individual tumors sprouting from the nerve sheath, okay? But it's really common. 1 in every 3,000 births around the world is someone born with the disorder, okay? Half of the people who are the first in their family to be diagnosed have a spontaneous mutation, and the other half are people whose parents had it too, but they were never diagnosed before. So about half the people are inheriting it. Half the people have a mutation that happened in the sperm or the egg that caused the disorder. Most of the time, as you see with the young woman here, the debilitating and disfiguring symptoms are not common before puberty, and then they often accumulate slowly. And so many of the patients have offspring. You know, when they're younger, young adults, it doesn't necessarily cause them that many problems. No one's ever been born. No one's ever been documented to be born with both of those NF1 alleles knocked out, okay? So, and the Mendelian inheritance risk for each child, if you have the disorder, to pass it on, is 50% for each child, although it looks like more than that is actually what's happening. So there may be functional changes in the sperm or egg that's making those actually more likely to be the ones that are fertilized. There's a lot of variability, okay, even within families that have exactly the same mutation. Some people don't have many symptoms their whole life, and other people are really seriously affected. Some of the symptoms are kind of present from the beginning that must be just due to having only one copy or only having half as much of the protein expressed. And other things are like the tumors themselves seem to be caused by a second hit that knocks out the good allele in that particular cell. Okay. Yeah, I mean, so it's called neurofibromatosis because these tumors are called neurofibromas because they grow on the neurons. Okay. But you can see there's a lot of areas of the body that are affected. Here's a friend of mine, Ashok, and he lives in Chicago. But he was born in Nepal and didn't have a lot of medical care when he was little. But you can see when he was small, he really did not show any symptoms. As he grew older, he started to develop one of these plexiform neurofibromas on his face, okay? And when you you cut away the tumor, you excise the tumor, it tends to grow back more aggressively, okay? Possibly because, you know, you're cutting it open, you're generating an immune response, which is, you know, releasing the histamine from the mast cells and so on. But a lot of times when they try to remove them, it grows back faster. And it becomes, you know, very socially and or physically debilitating. All right. This became a special interest of mine because in 2015, my son was diagnosed with this disorder. And he's the first in our family, like half of the people that are diagnosed. Oops, that's wrong. So I thought about what I could do as a parent to try to help give him the best possible outcome. And one of the things with the histamine, and I mean, the doctors say all you can do is watch and wait. We go to regular, you know, we'll do frequent checkups and see if anything emerges, and we'll deal with it when it does. That's not a really satisfying answer for a parent. So I wanted to pursue anything that I could, and one of the questions I had was, is there stuff I can do with his diet? Okay, so if you are histamine intolerant or have a disorder that histamine might affect, there are a lot of foods that actually have a lot of histamine in them. And then there are foods that actually cause your body to release histamine that is already stored when you ingest them or when you digest them. And a lot of them are healthy foods, right? And things that you'd want to, you'd really be interested in eating most of the time. So we wanted to know which foods, how much variability. One of the things they say is if you have a mass or a histamine disorder, if you're going to eat meat, you get the freshest cut of meat and you cook it immediately. And if you have leftovers, throw it away. Because even in your fridge, the bacteria will start to produce the histamine, and it's not something that goes away when you cook it. Okay. And so that's frustrating, right? And especially because there's really no good list of how much histamine is in each kind of food. Okay. And so we looked at these questions, and we had used some test kits that are actually provided for the seafood industry, because in shellfish, there is, really great risk of this, and you can get an anaphylactic shock just from eating spoiled shellfish because of the histamine levels. So we tried to, we tested a lot of foods. We looked at developing our own kit, and had a lot of success, but the actual enzyme
about Donald Trump.
The actual enzyme is not very cheap. And so we were looking into that when the COVID hit, and we were all shut down. All right, so then a paper came out in 2018 of looking at a skull that I was really familiar with already, Cro-Magnon 1. This is from an individual who lived about 28,000 years ago in France, or what is now France. And we looked at him all through grad school, and we have a cast of this in our teaching lab here. But he has all these lesions of the bone on his face, right? And so the question was always, what happened to this poor guy? And the paper that came out in 2018 was from a team of pathologists in Paris who had looked at it and done some CT scans and concluded that he probably had neurofibromatosis type 1. So this was really interesting to me that the disorder would still be, would have already been available or been in our communities 28,000 years ago. And I wanted to look into this further. So one of the main phenotypes that are associated with the disorder is macrocephaly, or having a big skull. They do circumference they take the head circumference at every appointment for these kids and compare it to the average. So 2 standard deviations above the mean is considered macrocephaly. So I looked at, we didn't have actual outside cranial circumference data from a lot of the early hominin, early modern human skulls, because a lot of them are broken, damaged on the outside, but there were endocranial volumes, or the inside of the skull measurements. And so I compared those as a proxy, and there was no real regional trends, but Cro-Magnon 1 is over on the far right, the second to the last one with the little star on top. And the other one is a upper Paleolithic skeleton from Spain that also appears to have likely had some sort of a genetic disorder or condition of some kind. So that was interesting. I also want to know, why don't non-human primates get the disorder? According to the literature at the time, the human and chimp genes and proteins were identical. But I talked to lots of primatologists, and there's never been a report of non-human primates having a tumor disorder like this, a genetic tumor disorder. There are some cases where they have like leprosy, that they'll grow some kind of similar tumors, but not an inherited disorder. And so why? So I was trying to, write about this, and I finally went back to the original source that people were citing, saying that the genes were the same for humans and chimps. And that source wasn't actually saying that at all. So then I got the genome data for reference sequences from humans and chimps for this gene. Actually, I looked at the protein because the gene is so huge. I thought, well, first I'll compare the protein, right? And it turns out the protein is not identical. And so, so nobody knew that before, and that kind of changed everything. So it's on chromosome 17. It's It's a big gene as far as genes go. It's got 300,000 bases in it, or 300,000 letters long. And of the people with the disorder, thousands of different pathogenic mutations have been founded throughout the length of the gene. So it's not just, you get one particular, genetic change and you get the disorder, that everyone shares. It's thousands of different mutations that occur. It's got a really high mutation rate. Okay, there are at least 10 functional domains, which makes it qualify as what we call a super gene, where there are different different genes being inherited together. Okay, and evolutionarily being packaged as sort of a unit. And there are 3 other genes embedded on the complementary strand. The best studied of those domains is what's called the gap-related domain, which is the one that is really clearly involved in the formation of the tumors. Which the function of it is actually to be like a brake. It downregulates what's called the RAS GTP, or the RAF-MEK-ERK signal cascade. Okay, it downregulates that, and that is associated with cell proliferation and tumor growth. Okay, so there hasn't been a lot of research into the other 9 domains, but it's very highly conserved. It is not a gene that's only found in humans, obviously, but it's not a gene that's only found in primates. It's not a gene that's only found in mammals. It's found also in the social amoeba. Okay, and so I wonder why. Why is it so easy to mutate and yet it's so highly conserved? So in the social amoeba, which is one that I'm going to be working with here in the lab, they found that in 2015, there's a paper that found that a lot of people are using the social amoeba. They also call them slime molds, but that's not really an accurate description of what they are. The social amoeba is used a lot in lab research because it's got a lot of overlap, or a lot of orthologs, overlapping genes, with a lot of neural issues in humans. Okay, and in the wild, they grow in soil and leaf litter, and they eat bacteria. But in the labs, they could grow them. They would find that they could grow them, colonies of them in a petri dish on agar, on nutrient agar. And somebody wanted to know, I wonder what the common thing is among all these various lab strains around the world that can do this. And it turned out that the mutation, that the gene associated with that is the same gene NF1. Okay, it's their homolog of our NF1 gene. And if they knock out the NF1 gene, then the cell membrane becomes able to ingest larger particles and larger qualities of liquid. And so then they can live they can grow in the plates instead of living in the soil. They don't have a RAF signal cascade like we do. So it's really directly affecting their plasma membrane. And they have interesting things about them. They switch from asexual to sexual reproduction, depending on whether they're under nutrient stress. And there's a lot of fascinating things that we might not get to, but that I'm going to be studying about them. Interestingly, C. elegans does not have a homolog gene. Okay, and I think that's interesting. Maybe they, their reproductive strategy, which is hermaphroditic, at times hermaphroditic, or often is, that may have made it not necessary to have this gene. Fruit flies, you see some of the things, if they have a full knockout, they have reduced body size, deficits in olfactory learning and long-term memory, and abnormal circadian rhythm and escape response. In zebrafish, there are actually 2 different homologs of the gene, and the phenotypes are observed when both of those are knocked out. But they have cardiovascular defects and overproliferation of oligodendrocyte progenitor cells. In the mouse, just like in humans, apparently, a double, knocking out both alleles, both copies, will cause the embryo to die. So all the phenotypes are observed in heterozygotes. But it causes overproliferation of astrocytes, okay, and a 50% increase compared to wild type. Deficits in memory and spatial learning have been observed in these mice. And they develop the neurofibroma tumors. sometimes becoming malignant. Okay. I talked to NF researchers who had tried to develop model lineages of non-human primates for this disorder, and they're not able to do that. They tried and they failed and they gave up. And it may have to do with this palindromic AT-rich repeat that they have and we don't, but we're not sure why. Anyway, there are a great variety of other ones that have of other species that have been studied for this particular gene. And I have data here, which you can look at in my one of my papers on my website if you want, which is that paper, the most recent one I presented in this spring. If you take the neurofibromin and you use it to as the basis of a phylogenetic tree of primates, they sort out in much the same way you would find if you just looked at a phylogenetic tree of that was based on the genome of primates, which is really interesting. So it may actually be an important factor in speciation. In humans, I keep checking this to make sure, but no modern human has ever been found to have the chimp type allele. I mean, okay, so nobody is living today that was born with the chimp type allele, even though it mutates so readily. The gene is not, is not producing a change, a survivable change apparently in that particular codon. The difference is in a relatively weak phosphorylation site in a region with strong phosphorylation sites. And so it's predicted to elicit, compared to chimps, a slight increase in overall phosphorylation potential and a decreased affinity for certain kinases. So I looked at ancient DNA that was available. And for all of the Neanderthal, Denisovan, and early modern human skeletons that had genome data available, they all have our modern human type, not the chimp type. So the human type allele appeared before the split of Neanderthals and modern humans, and it has become fixed in our human population. These are just some data that we don't have time to look at, but interesting. I mean, this is showing homozygous and heterozygous variants in the different across the gene compared to the HG9 human reference sequence. And so you can see that there's a lot of homozygous variants, the blue ones, in the Neanderthals and Denisovans. This early modern human has a lot of heterozygous, one copy is different than ours. And then you can see there's a lot of variation in modern human populations across the genome, but none of those variations are in that particular one that separates us from chimps. Okay. Which time we got? Maybe 10 minutes. Okay, so my hypothesis then was that maybe there's some kind of an adaptive advantage to having a big gene that affects lots of stuff that mutates really easily. Okay, why would that be adaptively advantageous? Well, maybe it's not at the germline level. Maybe it's at the somatic level. Maybe it allows your body to take advantage of mutations in that gene when you need a new neuron, okay, or when your neurons need to be able to grow farther. So you're thinking about in the way it affects the social amoeba, that it makes their cell membrane able to do more. And so this was a question I wanted to pursue more. Here's my son's back. These little birthmarks, a lot of them are oval, they're called cafe au lait macules. Having a lot of those in a child is usually the first indicator, and that was for us. And there's a lot of things affected, okay, that I know from studying human evolution for so long that are things that are important in human evolution, and that those are traits that we talk about as being important to follow when we're looking at human evolution or hominin evolution. But I got really interested in these cognitive ones at the end here, okay? So attention, behavior issues, spatial reasoning, Executive function disorder, like you know what your end goal is, but it's difficult to know what steps to do or to figure out what steps you need to do in order to achieve that. Difficulties with prosodic interpretation. So maybe they can't tell when people are being sarcastic as easily as a lot of us can. And challenges with speech and musicality. Okay, one of the studies that came out of Brazil was that they found that people with the NF1 disorder actually have amusia, or they have a hard time with music. There are also reduced risks of addiction, diabetes, and obesity among the people with the disorder, which is interesting. So I looked at, as a model for how to study this, I looked at the FOXP2 gene, which had been identified in 2002, and it was one of the things we talked about a lot when I was a graduate student and followed. It was identified because it's required for the normal human speech and language abilities. It was because a family in the UK had a lot of members that were born with this, the gene was identified because they had a hard time with speech and language, and they wanted to know what was common in this family, and it was turned out that they had a mutation in this FOXP2 gene. And the chimps form of the protein is different in 2 places from the modern human one. So I used that as sort of a comparison, did a whole study looking at just the literature for that, and thought that it was pretty interesting that we had these 2 big genes that are involving a lot of things that have to do with our ability to like transmit culture and our ability to do kinds of attention and focus and do spatial activities, right? Like navigating across an ocean or across a landscape. And so I thought, how interesting, right? Also, the NF1 gene is much, much older, okay? It comes from much earlier. The FOXP2 is only found in vertebrates. NF1 is found in the social amoeba. So those are some questions that we asked. And one of the things that we found was that the common denominator for ancient peoples are people who lived in cultures before we had written language. And we know this from indigenous peoples who still have a lot of knowledge of how these oral traditions work, is that they would encode their information through art and music. They use art, music, performance, stories, and features of the landscape as mnemonic devices to be able to encode like huge, vast quantities of information that you need to survive. And so this, was really an interesting connection. It's a correlation, not causation, right? And so we looked at, I had someone who's an expert in the oral knowledge in studying comparative worldwide oral knowledge systems, make a list of the skills that you'd need to be able to do this, or that a community would need to be able to have a knowledge system like this, because evolution acts on groups, not on individuals. And then I assessed which of those skills that she determined were actually challenge areas or strengths among people with the disorder. Okay, so if you break one copy of the gene, how does it affect those skills? And I assessed which of those skills. Some of them chimps do too, so those aren't really relevant to human evolution, right? Because they're already they're nothing new that's being produced. And here's just a summary. But some of the interesting things is that in people with the NF1 disorder, some of those skills are actually things they're better at. Okay, and probably people with a lot of different neurodiversities would be better at certain elements of this list of skills that it's needed. Yeah, so that's a whole other talk for a different time, but it's interesting stuff, and it really supports the use of the creative arts in teaching and learning, and in looking at neurodiversities in different ways, both in the classroom and in the workspace, I think. Oh, what have I done? Some weird thing. Okay, we don't really have too much time to talk about this, but this is the project that I'm starting on now. I never had a lab that I could actually do the DNA work in, and now thanks to my friends here in biology and our dean's office worked together to get me a space here that I'm just moving into now here on the second floor Wagner. And so I'm going to be looking at the social amoeba for a variety of different kinds of research projects. And I like it because it's cheap, they reproduce quickly, and they can answer a lot of different questions. So we're going to be looking at, environmental characterization. So the kind of soil questions I was doing before, but now let's see what amoebas are living here and make a phylogeny of amoebas around the world, and see what kind of information we can get for those. And we're also going to look at genetic, epigenetic, transcriptomic, proteomic, different questions. So comparing what the gene is and then what's being expressed, those kinds of things. And then also I got a portable sequencing device, which just came out recently. So you can take it in the field and you can like process, you can extract your DNA and you can run it off a laptop. They're doing stuff in the rainforest, studying different kinds of genetic differences in, I think, animal dung. But anyway, so we got one of those and we're going to be starting to optimize to use that. But it's a really interesting organism, and it lives everywhere. So we can go and collect samples around Macomb or wherever we want. It's got 6 chromosomes. It has a lot of trinucleotide tandem repeats. So we can create something that's similar to the short tandem repeat technology that's used in forensics for human DNA. And then students can do kinds of, the similar kinds of research based on that. I won't go into all those, but there's a lot of information available. Like I said, they do reproduce sexually and asexually. Oh, they have 3 sexes. And then when they do produce sexually, they can only reproduce with someone that's not the same as them. And there's a lot of different stages of their development, as you can see here. So we can isolate different stages and look at what's happening in the gene there. Just to finish up quick. Oh, I'm going the wrong way again. So we're going to be doing these projects. I'm going to be getting different students that are interested in different kinds of different areas and do semester-long projects on these. And I already collected a bunch and have them in the freezer. But that's one of the really great things about them, is that they have the ability to. Oh, I'm just going to throw out time. Kind of overview those anyway. They're quick and inexpensive. They're right in our backyard, and they can be frozen to pause research. So you can leave for winter break and put it in the freezer, and then you can take it back out and they'll start going again, which is like really fabulous for people that are doing semester-long projects and me not having to come in and deal with the colony on all the breaks. So, oops, wrong. All right, so the next steps then, I'm moving into the lab here. I have funding and equipment for the original research with these dictyostelium colonies. And if anyone's interested in talking about adding a forensic biology option to the undergrad major, biology, the bachelor's major here, I think it would be really easy to do. And I would have a lot of projects that people could work on. So there's my email. Again, everything that you looked at is in more detail on my website at halvashear.com. Does anyone have questions? Any question? All right, well, thank you all so much.
If anybody needs to go to class, I understand.
Yeah, something you're talking about you're looking at the
neurofibroma.
Yeah. As being very mutable, like it's a long gene. So do you think that there's more mutations arising there, or it's more tolerant? That fewer of them are lethal, or in what sense do you mean more mutagenic?
Well, it has a very high mutation rate. Like I said, 1 in every 3,000 people is born with a protein.
Kind of changing their amino acid sequence.
Well, yeah, I mean, as soon as it makes that protein not do what it's supposed to do, not be the proper sequence, then you have the disorder. Okay, and so it does have a really high mutation rate, and yet it's been conserved for so long. So I think that you know, as an evolutionary biologist, that's a really interesting and weird thing. And I think, and the fact that when you use the protein sequence to create a phylogenetic tree, it actually separates them out by species really, really well. Within the apes, and it only didn't separate chimps and bonobos, okay, which are two species of chimpanzee that split after we did from the chimps. But it split everybody else into their different species. So that's pretty fascinating, I think. And it suggests that there is a relationship between that particular gene and speciation events. Possibly changing something in the, you know, the sperm or the egg membrane or something that makes it possible to create new species. Hypothesis, not proven, but yeah. Anything else?
So can I follow up? So are you suggesting that there are other genes that if you study them, do not sort of spit out the primate phylogeny that we expect?
Well, so looking at the FOXP2 gene, it doesn't. In that gene, the language associated gene, there are only two differences among all primates. So this is something different than we see in most of these big genes. Anyway, I started studying it because my son had the disorder, and it just got more and more and more interesting all the time. So, so yeah, I mean, who knows? There's always new things to learn about it, apparently. And I'm excited to have a lab where I can explore some of those, you know, in the laboratory. Thanks all for coming. Appreciate it.